All right, let's start with the audio check. If you're watching the stream and you can hear me or you can't hear me, which is more difficult, of course, let me know in chat because then we can start. Like, I'm excited. I'm excited. So, been having a holiday, so sitting at home doing nothing really. So I thought it would be good to do a live stream, and I'm wondering if people can hear me. Misha, are you there? Can you hear me? My microphone is doing like the audio thing, but I'm still getting this YouTube errors, so I might not. No audio. Let me just put it in chat so that people can answer me if there's audio. If not, then there's no audio, which would suck badly. That's so annoying. Alright, let me see what's going on here. Okay, so my microphone should be on. Huh. Gonna have to check on my phone just to make sure. Gonna have to check on my phone since no one chat is answering, which is okay. Like, you don't have to put anything in chat, but let me see, let me see. Let me open up the sound. Yes, but the sound is a bit low and hollow. Yeah, that might be the case. I can put it up a little bit higher, but I'm using a different mic. So I can put it a little bit closer to my. But yeah, that's always the case with live streaming. And since I'm doing it from home, I don't have the equipment that I have on my work. So that's just a shame. But I hope it's audible. And in the end, it's just about the programming, right? So I think people will enjoy it. And it's not so much about my face or how I look. But let me show you guys the. So everything here, what we're going to do is just like we're going to use open source databases and tools and things that are available to analyze some monkeypox data. And why monkeypox? Well, it's hip and happening at the moment. So that's the reason that I chose monkeypox. You could do it with any virus or other genomes. But I just thought it would be fun to give you guys an example on what you can do with it. Because like I've been doing the R course and the bioinformatic course, and people always wonder or people always ask me like, you give us these little examples to practice on, but we never see you making something. And of course, making something bigger is something that's difficult when you're just alone. So let's just start, right? So I'm going to open up my notepad window. I am going to open up myself. I hope. I hope, I hope, I hope. No, this is all wrong. That is so horrible. All right, so let me switch that to the correct screen because I want to use monkey. All right, now you can see me as well. So hoping that the audio's fine because that's one of the drawbacks. Um, don't have the equipment. But good. So for today, this is kind of what we're going to do. So if you want to follow along or if you want to do the coding yourself, I made a GitHub repository. So let me put the link in chat. And I will be pushing my code there while we are doing the analysis so that you can kind of see how I would normally work on a normal workday. So here's the live stream. So this is the GitHub. So it's github.com, my username, and then monkey2022 or monkey22. Good. And with that, like in the end, we need to start somewhere, right? So I made a little idea of what I wanted to do today. So the first question is where do we get our data from? And that is, of course, a tricky question. There's a lot of sources of data out there, but since monkeypox has been recently kind of rediscovered or discovered in the UK, but also in Portugal, Belgium, and the USA, there's a lot of new sequencing data coming out. And most of the new sequences I found on virology.org, which is a website where people share virus sequences. And you can kind of publish little articles there on genome analysis. So that's where we're going to get our data from. And of course, we're going to use NCBI for known previous monkeypox outbreaks and also other pox viruses. Because in the end, when you're doing phylogeny, you're not just doing phylogeny on a single genome. You want to kind of put that into context. And then we're going to load the genome. We're going to create a BLAST database. We're going to load the associated proteins from NCBI, which is one of the issues with new genomes coming out. And this is kind of the reason why I wanted to make this. I wanted to show you guys how do you analyze a genome when there's no information about that genome really available except for the ACTGs that you kind of find on a website, right? And then because we need to create our own protein models, we can use other pox viruses like smallpox or the earlier monkeypox outbreaks to kind of combine this information from these different sources or from the different protein sources together to create protein models. So we're going to use Levenstein distance or multiple sequence alignment to do something like that. Then we can create a consensus sequence, and then with this consensus sequence, we can finally start annotating the new genomes that have been published. So I'm really excited about that. I haven't seen any work yet. The only thing that I saw is that people just take the whole DNA sequence and just do some multiple sequence alignment on it, which I don't think is the best way to look at viruses. Especially one of these monkeypox virus where you have two of these like hypervariable regions where genes are duplicated and there's like different versions of the same gene across the genome. And then in the end, I wanted to show you guys like a little picture of what you can do. But with that out of the way, I think the first thing that I wanted to show you guys is how to get your data, right? Because if you're like me, you're not a virologist, you're just a basic bioinformatician sitting at home doing nothing, worried about an outbreak, then the first thing that you do is try to get some sequence data. So for that, of course, we need to start up our trusty Firefox window, and that goes wrong again because for some reason Firefox also switched to the wrong window because I don't want to show you guys my live streaming window. I just want to show you guys my empty Firefox window, and then we're going to go and put it away. All right, so what are we going to do? Well, we're going to go to NCBI, right? So NCBI is the National Center for Bioinformatics, and we are going to go to the nucleotide core database because we want to download DNA sequences, right? So then you get onto a website. It's a little bit big, which looks like this, right? So you have the word nucleotide because that's the database that we want to search in, and then we want to search for something. So we're just going to be like filling in monkeypox, right? Because that's what we want, right? So, and then it comes back and it says, well, there's a monkeypox virus, which is a double-stranded DNA virus in the family of Poxviridae. And then here we have, for example, the recently sequenced USA more or less genome. Right, so if we click on it, then we see that it's a big sequence, so it will download. What we see is a whole bunch of information. So it's the monkeypox virus isolate. It has a name, and this is the complete genome. Here is the GenBank ID, which we're going to need. And this is .2, so it means that it's already version 2. So the Americans published their first draft very soon from their first patient. I think only like 3 or 4 days after the outbreak. And then they decided, well, we are going to sequence at higher depth, and with a higher sequencing depth, it means that they have more coverage and that their genome is just better. So you can see that the virus is relatively long. It's 197,000 base pairs long. It's a direct submission, and here we see some more information. So we can see that it's a monkeypox virus. It's genomic DNA of the virus. This is the isolate. So that means that this is the more or less patient ID from which they got their virus sample. And the host was Homo sapiens. So that's humans. And it's from USA. MA stands for Massachusetts, I think. Correct me if I'm wrong, but that doesn't matter too much. In the end, we're just going to more or less collect a whole bunch of these viruses. So how are we going to proceed? Well, we're going to proceed by just putting up an Excel file. So this is an empty Excel file that I created. Well, there's nothing in there, so I'm just going to say I want to have a column which is called ID, right? And that's going to, I'm going to use that. So I'm just going to copy in the GenBank ID from the USA monkeypox virus. And then the next one is going to be, well, what kind of species is it, right? And then the host. And then we want to know the region. So in this case, it's North America. And then the country, which in this case is USA. And the host was Homo sapiens. And the species, let me copy that from here because this is an orthopox virus. All right. So this is how it looks, and this is how we're going to treat our data. And now comes the boring part, right? Because now we have to go here, and then, okay, so now we got the first from the US, right? We saved our GenBank ID, and then we're going to go to the next one. So we're going to just click on, for example, a different one, and we're going to see. So this is an unverified monkeypox strain from the Democratic Republic of Congo. This was not found in humans. It was found in another animal. No idea what animal this is, so let me look for that. So it's kind of a mouse kind of thing, Spitz mouse. So yeah, it's a mouse. Um, and we can download a whole bunch of these. Again, you see that it's just the DNA sequence, right? And even worse here, there's a gap because they couldn't sequence the whole thing. So we see here that there's a little gap. But what we can do, of course, is find one which is annotated, because we have previous virus outbreaks where we do find nice annotation where there are proteins annotated. So let me see. Yeah, so here we have one, and here we can see that they have annotated genes and coding sequences and genes and coding sequences. So we also have the protein sequences here. But of course, this needs to be translated to the new outbreak, because it could be that there's a lot of difference, right, between the two. Um, Genome might be longer, there might be massive gaps and these kinds of things. So in the end, hey, you have to prepare your Excel file, you have to add a whole bunch in, and of course, because I'm a good streamer, I already prepared some. So I downloaded, or not so much downloaded, but I took my time and I just selected a whole bunch of these pox viruses. So in total I have like 75, and then I thought, oh, that would be enough. So we have IDs, we have the organisms, which is all orthopox virus. I put in some other species. So these are goats. We have here some birds. We have a salmon, which is a fish. And for the rest, I also put in some cowpox. I also put in the Acmeta virus. The Acmeta virus is very closely related to the pox viruses. It's in the same group. But it's not a standard pox virus. I added a couple more, like volpoxes and raccoons, and of course every time you have to go to NCBI, look up where the sample was collected, what species it was collected on, and sometimes you can't find this information. I also put in the vaccine. That's the vaccine that's being used for birds. And besides that, I also think I put in some of the really bad ones. So these are the variola strains. So that's smallpox, right? So they are different. But in the end, we have this big Excel file. We spent some time on making it. And fortunately, I didn't have to make this one myself completely, because fortunately on virology.org someone actually shared a very small subset of what they were looking at. So they think it was the Belgium group which published their first genome sequences, and then they gave a little table like this, a little bit different. They didn't split it up between continent, country, and state. They just had only countries, and they only looked at humans, which I think is not what you should do. Right? If you compare viruses, viral genomes, they can be very different between different species. So you always want to get a couple of outgroups, and these outgroups are very important because they kind of give you a way to anchor your virus into this virus tree. And that's why I always put in some goats, some cows, some other animals, so that we can compare. So how do we proceed? Well, we just select everything, Control A, we copy it, Control C, and then we go back to Notepad. And in Notepad, we just create a new file, and we are going to save this file. So when I copy paste, it's a separated file using tabs. So I'm just going to save this into my monkey, and I'm going to say monkeypox annotation.txt. So that's how I'm going to call it. Oh, you guys can see that I'm saving it, but I called it monkeypox underscore annotation.txt, and it's in the data folder. So the first thing that I'm now going to do is going to commit this file to GitHub. So I'm going to say git add data slash monkeypox underscore annotation.txt, and then git commit -m initial data file, and then I'm going to push it. So if you go to the GitHub repository, which is in the chat, so github.com, then monkey 22, then now you will see this file being there so that you can use it for yourself. Good. So of course, where do we get our data from? So I just got a whole bunch of sequences from NCBI first. I'm not going to deal with the newly published sequences because fortunately a couple of the newly published ones are already in there. So we have 2 from the US, version 1 and version 2, and we also have the one from Great Britain, which was sequenced a little bit earlier, but has still version 1, so it's not a very up-to-date sequence. But we have 2 of the new monkeypox, and we can now also start looking at what is different between the first sequencing run from the Americans and the second one. But first things first, we go to our code, and we're just going to say we're going to read in this table, right? So I'm just going to do read table. I'm going to load in data slash monkeypox, annotation.txt. My separator is a tab. I am going to say that I have a header because I added one, and my row.names is 1 because I want to use the Ensembl IDs as the row names. So this should load in our file without any issues. And how do we want to call this? I'm just going to call this source IDs, right? Because I need to get my IDs from somewhere, so I'm just going to get them from a table. All right, so let's quickly go to R, see if that works. So then I have to, yeah, that works. So let's go to R and load in the data file. I also have to load the libraries. So you can ignore the API key.r. If you query NCBI and you do that a lot, then it's better to have an API key so that they can keep track of how many requests you do. You can do everything without an API key, what we're doing here today. You just have to make sure that you're not overloading their server. So if we look at source IDs, then this is what we loaded in. So a whole bunch of different viruses, a whole bunch of orthopoxes, some monkey 22s, and a whole bunch of older ones as well. And here the table continues, so that's all fine. So in total we should have 75, so let's check that we really have 75. So we do have 75, which is what we wanted. Good. All right. First things first. So where do we get our data? We just go to NCBI, we make a little Excel table, and now we want to download this data, of course, because we now want to get access to the genomes. So how do we get access to the genomes? Well, getting access to the genomes is really useful because is really simple because we have this rentrez function, which allows us to automatically query rentrez. So how does this work? So it works more or less like this, and I'm not going to use the API key first. So let me show you guys Notepad++ again. So what I'm going to do is just say rentrez underscore fetch. So I want to fetch from the nucleotide database. I want to fetch an ID. So fortunately we have an ID because we have the row names of source IDs, and we're just going to use the first one, right? So just download one sequence just to make sure that it works, and I'm going to say I want to have the Fasta sequence back because I love working with Fasta files. All right, and then I'm going to say mdata. right? So my data, just first to check. And then we're going to go to R, and we are just going to do the query. And what I'm expecting is now that I get a long sequence back. So if we look, then indeed we see that we get like a whole bunch of sequence data, and it also gives us the FASTA header saying that this is indeed the USA genome that we just looked at. This is actually version 1. We could download version 2 as well. The nice thing about the rentrez fetch function is we want to get the first 2. We can do the same thing, so we can just say 1 to 2, and then we get 2 sequences instead of 1. If I look at mdata, it still shows all on one line, so it kind of flips the R window around because it's too much data to deal with. But what we can see is that there are /new lines in the file. Right, so the first thing that I'm going to do is say, well, I don't want everything on a single line, right? I want to make sure that I have my return sequences and that I'm going to split them. So let me just call this rsec, right, for return sequence. And then what I'm going to do is I'm just going to str split the rsec, and I'm going to split by new lines because that's kind of how a normal person would read it. And then I get something back which is readable. So now I have like 60 base pairs per line, and there should be header lines in there somewhere, but the virus is so long that I can't even scroll up to see what's happening. So I'm just going to take this little piece of code, and then we go back to Notepad, and we're just going to add that, right? So I'm going to call this return sequence, and I'm just going to query 1. No, let's query 2, right? So then we have something to work with, and then we can do some data analysis on that. So of course, because I did a string split returns a list, so I'm just going to unlist it because I don't want to deal with the fact that it might have multiple elements in there. So just unlist it, then everything's on a single line. So if we now would look at the first 10 lines of this, then it looks somewhere like this. Notepad, let me show you the R GUI again. So let's look at the first 5 lines, right? So we see the first header line, and then we see the code that belongs to it, and somewhere in the middle there will be a secondary header line because we asked for 2. So let's figure out where those header lines are, right? Because in the end, I want to have all of the sequences in a vector, and the names of the sequences are going to be the names of the vector, right? So I'm just going to kind of combine the 2 data sources because now it's in one vector. Sometimes a line means it's a header, sometimes a line means it's code. So how are we going to do that? So back to Notepad++. So we do an unlist on this, and then I'm just going to override my rsec because that's how I roll, because I don't need it, right? So first I want to figure out where are the header lines. So I'm just going to say grep. And what am I going to grab? I'm going to grab the larger than symbol, and I'm going to do that in the return sequences, right? And this should give me back which lines contain the header. So let's go to R, and now when we look at header.lines, we see that we have one header line, which is, oh, I didn't override it. So I looked at it, but I didn't put it in rsec. So let me make sure that everything's okay. So I'm going to do the query again. And we have our header lines, and now we should have 2 positions, right? So the first line is the header line, and then the next header is at line 2820. Good, so now I can figure out where my sequences are, right? Because my sequences go from line number 2 to line 2819. So the last one is going to be from 2821 until the number of lines that I have. So how do I build this up? Well, I know that my start is going to be, so let's go back here. So my start position is going to be the header.lines +1, right? Because it starts at the line after the header. And then my stop is going to be, and here I need to kind of think a little bit, right? So I need to throw away the first header line, right? Because the first header line was 1. So, and if I would just say header lines, because normally it stops at header lines -1. But now it would get 0 as the first entry, which I don't want. So I'm just going to say, from the header, throw away the first header line, because I don't need that one to figure out to stop. So what do I need to do more? Well, I also need to add the total number of lines that I have at the end, right? So I'm going to say length rsec, and now if we would go to R, then R would be able to tell us that the start of the header is going to be at header lines +1, so that's at 2 and at 2,821. And the stop is going to be at 2,819. So the first sequence goes from here to here, and the second sequence goes until 25638. Good. So let's cbind these two things together, so to get a little bit of a table. So I'm just going to say, instead of calling start and stop, I'm just going to say cbind start is this, comma, stop is, and then I'm just going to take the stop positions. And I am going to call this sec.pos for sequence positions. Right, so I do a grep to figure out where they are. So let me add some comment, Fasta header, and the lines at which the sequence starts and stops. Alright, so now I have my sequence position, so now I only have to do one thing, right? I just go through this little table that I made. We now have 2, but if we would download 3 sequences, we would of course get 3 positions. So let me show you guys in R how that works. So we download 3 sequences now. I just said 1, 2, 3. And then here we have our header line, and now our sec.pos is a little table which says that the first sequence starts at 2 until here, to there, and the third sequence starts from here to there. Good. So how can we now use this, right? So we can just say lapply to the sec.pos to the first to the rows. I want to execute a function and function of x, right? And just so what do I now want to do? Well, I want to get the sequences, right? So I just want to say, well, from my return sequences, right? What do I want to get? Well, I want to get a sequence from the start, which is x start. And I want to have this sequence run until the x end. So, and this will create a vector containing 2, 3, 4, 5, 6, 7. And I need to make sure that they are actually numeric values, because otherwise R will just randomly take the character values from it. But I'm just going to say as numeric just to be sure, right? And now I have this little sequence, right? And this is just from the beginning until the end. I just name all of the rows that I want to get. So what I'm going to do, I'm going to, from rsec, select these rows, or select these entries, and then I'm going to paste them all together, and I'm going to say separator is nothing. And to make sure, I'm going to say collapse is also nothing. And these are going to be my FASTA sequences. All right, let's go to R, see if everything works, if I typed it in correctly, and I did not. So it says unexpected closing, so I'm going to go to Notepad and I'm going to use code highlighting. So here I have a closing, which I don't want because I want a sequence from the start until the end, and then I want to use this as the selection to my return sequences, and that should all be fine. All right, so let's go back to R, and we should have fixed the Alright, match.fung, all is not a, one is not a function. Ah, right, yeah, because I need to not use lapply, I need to use apply, of course, because I'm dealing with a matrix, because sec.pos is a matrix. Good, so a little bit of debugging. 2 must be a finite number. Why is 2 not a finite number? Should be, right? Sec.pos, all finite. Sec.default. Alright, so let's build in a little bit of debug, and then I'm just going to say cut x start, comma, space, comma, x, end. And put a new line behind it. Alright, let's go to R, run the code, and it goes from 2 to NA. And that is because it's not called end, but it's called stop. Little errors. We'll encounter more, right? So we go here and we call it stop. And we call it stop here, and then we delete our little line of debug, and then we're going to go to R and make sure that it works, right? So now when we look at our sec.fasta, we should have two FASTA sequences. So I'm just going to lapply to the FASTA sequence and char, right? Because just to know how many characters there are. So you can see that the first American build from the monkeypox genome had 197, 128. The new one has 4 base pairs less, and then this is another monkeypox virus, which is the one from the UK. So that's the Great Britain one, which is a little bit different in length again. But at least we learn that the monkeypox is a very, very big virus, and this very big virus is having a lot of sequencing data. All right, so back to Notepad++. So we still need to do something, right? Because we still need to put the names on the FASTA sequences. So I'm just going to say that the names of sec.fasta is going to be the rsec, and then I'm going to take the header lines, and those are going to be my names. So if I go to rgui just to check, so now if I look at sec.fasta and I look at the names of them, I should get 3 names, and indeed I get 1, 2, and 3 names. Perfect. All right, so that is our first challenge done. So to do one is load genome sequences from NCBI and FASTA. We can do that using this, but I'm just going to make a little function. So I'm going to say load genomes is a function. It takes IDs, right? So IDs, and I'm going to return our sec.fasta formatted at the end from this function, and instead of using row name sequence IDs 1, 2, 3, I am just going to swap this out and say IDS. Perfect. All right, so we create a new file, and then we put our function in, and we're just going to say save this, and we are going to put this in R, and this is going to be load genomes. Let me call it download data.r, right? Just a little bit of a different function name. And I'm going to here also call this download data. Well, not .r. All right, so we have our first function. So the first function is just going to download data, and I'm just going to do the first 3 just to make sure that I have it, that I tested, right? Because if I would download all like 75 sequences, then I would stress the server a little bit, and if it would go wrong, I need to do it like 2 or 3 times, and I don't want to do that. So I'm just going to call this NCBI sec, right? So, and I need to also do a source because I need to load in my function. So I'm going to say r/download data.r. I'm going to source this file. It will contain my function, and I'm going to query the first 3 sequences. All right, so let's go to R, make sure that everything works and that we get our 3 sequences. So now when I look at ncbi.sec and I ask for the length, I expect it to be a length of 3 because I asked for 3 sequences, and the name should not have changed. Good, so this seems to work, so I'm now going to remove my artificial imitation and just download all 75 genomes in one go. So I'm just going to press enter. It will take a little bit of time, but that should download all of them, and then we can check if everything went well. All right, so let's not wait for R. Let's just go here. Right, so I'm just going to remove it. So very clear code, like we have a single file that we source, the code is there, and then we have the function call. All right, Perfect. So R actually finished. So if I ask for the length of ncbi.sec, I have 75 sequences, and I could ask for the names as well, and then we would see that there are names. So we get a little bit of information, right, where they're from, but they're all coded. That's why we make the Excel file. The Excel file is our annotation, which we're going to use to make trees or do other things. Good. So the first thing that we need to do is make a BLAST DB, right? Because we now have all of these sequences, but we need to do something with these sequences. And we need to annotate them. So annotating these sequences means taking a protein sequence and then blasting it to our BLAST database. So let's make a BLAST database. So fortunately, a BLAST database is very easy because a BLAST database actually takes a Fasta file as input, right? So we can just write out our Fasta file. So we can just say for header in names of our ncbi.sec, so our NCBI sequences that we just downloaded. What am I going to do? Well, I'm going to cut, and I need to figure out where I want to store my database. So I'm going to say that database is located in data slash DNA db.fsa. Right? I'm just going to give it any extension. Doesn't matter where you save it. So we're going to cut, and we're going to cut to file is our database. And what do we want to cut? Well, we want to cut our header, and then we want to add a new line to the end, and then we are going to cut our sequence that we got. So we're just going to reconstruct the stuff that we got from NCBI, and I'm going to say append is true, so that actually we don't overwrite the file every time, and that means that I also have to make the file. So I'm going to say cut nothing, empty string, into our database. So file is db. All right, so just rewriting out the data that we got from NCBI to a file on my hard drive. This also means that I don't have to continuously reload the files from NCBI, because once I have the database, then I can just use my database. Good, so run the code, runs really, really fast. Let me open up the database for you guys so that you can see it. So let's go back to Notepad. We go to C, Github, Monkey 22 data, and then we do our DNA database.fsa. Right, so here we just see the database that we just created. So again, the names with the sequences, and of course the sequences are on one line. Fortunately, BLAST doesn't care about that. The Fasta convention is to have 60 characters on a line, but we don't have to. All right, so again, creating the BLAST database. Let's make a little function out of it as well, right? So to keep in the structure, so I'm going to say function. So make BLAST DB is a function which takes as an input sequences, and going to add this, and in the end I want to return the database location where I stored it, and I want to also make this variable because we might want to make a database with protein sequences as well. So I'm just going to put database as an option, give it a default value, and now I have to make sure that I replace all of the NCBI secs, so the NCBI sequences, with the sequences that have been put in. Just in case we might want to download 2 or 3 different NCBI sequence databases. Good, so now we have our make BLAST database. So I'm going to, of course, call this with our ncbi.sec, and I'm going to store this as database so that I know where the database was created. Good, and I'm going to make a new file, put the data in. I'm going to close the monkey annotation, and I'm going to save this in R, and I'm going to call this blastdb.r. All right, so now here we have to source it, and we are going to source blastdb.r, and then we have the function available, and we can do it. Good. Any questions so far? We downloaded sequence data from 75 different genomes. We made our BLAST database so that we can BLAST. And we now go and do the next step. And the next step is going to be load our associated proteins from NCBI, right? Because we want to get protein models and we need to get those that belong to the virus. So there's two ways of doing this. We could just go to NCBI and start doing the same thing as what we did. Just make a new Excel file, put all of the different proteins in there, but we're not going to do that because we're going to use this rentrez function again. Right, so this rentrez function is a very, very useful function or very useful package because it allows us to do cross-database queries as well, right? In the download data, we just do an rentrez fetch, but rentrez also has an rentrez link function. So the rentrez link function looks a little bit like this. So we can do rentrez link, and then we can say db from is. So we want to search from the new core database because we had the nucleotide database. I want to say that the ID that I'm going to search from is the one that we found, right? Because when we looked into the Firefox window, I think that's this one. Firefox, where are you? We found that this one here, right, has proteins annotated. It doesn't just have the DNA sequence, it does, that's all the way at the bottom, but it also has proteins associated with it. So just let this Zaire 69, 96, so it was found in Zaire in 1996, and I'm just going to take this number, right, from here, and I'm going to go to Notepad, and I'm just going to say, well, I want to get everything which is in the nucleotide database attached to this nucleotide database ID, and I'm going to say I want to get everything which you have in your protein database. Right, so I'm just querying from the nucleotide database asking which proteins in the protein database are annotated to this sequence. And I'm going to call this links, right, because it's called rentrez link. All right, so how does this look in R? So if we go to R, if we do an rentrez link, then now we get an object, and this object is pretty complex, right? It's an E-link object with contents. It has links, so the IDs for the linked records from NCBI. So how do we get now the protein sequences? Well, we can ask the str to look at the structure of this object, and then we see that, hey, interesting, it has something called $links, and that's a list of 2, and there are the new core proteins in there. And we also have the coding sequences. We don't really care which one of the 2 we take. So let me see how that looks. So we say links, $links. Then we get this, and then the links that I'm interested in are the new core proteins. And these are the IDs for the proteins which are associated with this monkeypox virus which was detected in Zaire. So, and let's just copy this and we go back to Notepad. So in Notepad we're going to say that these are the protein.ids. And now of course we need to make sure that there are really proteins, right? Because if the length of this is larger than 0, then of course we have proteins. Because, and let me make sure that that is correct. Let me open up this monkeypox annotation, because when we looked at the original data from the American group, we saw that there were no links, no protein links. Right, so if we would just query this American sequence by just updating the ID, then we would not get back anything. Right, because now if we look at our links object, it will still say that it's an E-link object. But when we look at the links, it's null. There are no protein links which are associated to this virus. So in this case, we can't use the American sequence because they have not annotated their proteins yet. But we can use the Zaire virus to get all of the proteins. In theory, of course, we could go through all of the 75 entries that we have, see if there's a protein associated with the virus, and then remember that. But that's going to be 75 queries to the database, and we don't want to stress their database. So I'm just going to say, well, let's just do one first, right? No, let's not do 75 queries and download hundreds and hundreds of proteins, right? Because you can see that with one genome sequence, right? So from this one genome sequence, there are 191 proteins annotated on the genome. Display is not clear. Oh, is that so? Does it need to be bigger? Let me do an edit. GUI. I can make the GUI a little bit bigger. I hope that that's more clear then. Is this more readable? Just hoping that it's good. I don't see any issues. It might be my streaming quality. I'm streaming from a laptop since I'm at home, so I'm not at work, so I don't have the like fantastic internet that the university normally gives me. But we'll just have to do with it. And of course you can get the code from Github. So I'm just going to write down this function and then we're going to commit the functions that we had so far. So you can follow along and see it high definition there. But what we see here is that when we query a nucleotide sequence which does not have proteins associated with it, we just get a null back. And that's not what we want because we want to get the protein sequences. But like this should be okay. And I need to see my streaming. All right, so if the length of the protein IDs that we get is larger than 0, then we get proteins, and now we want to download the sequences. Fortunately, we already wrote a function for that, right? We wrote a function called download data. But the function download data, if we look at it, it actually has the database that we want to query hard-coded. So I'm just going to say here, db is, and standard, I want to query nucleotide, right? And here I'm just going to change the word db or the nucleotide by the db that we specify. So now here in my analysis, so if the length of the proteins is larger than 0, I can now just use my download data. I can give it the protein IDs, and I can of course now query the protein database. So I'm just going to update that, right? So let me make sure that I actually reload my function. So let's go to R, reload the function. So now when I look at download data, the function actually now has two parameters. So the IDs that I want to query, and then the database I can specify by the second parameter. So that should be okay. And of course, we get our FASTA data back, so we also want to have the proteins in FASTA. So when we go to Notepad++, we can just say, well, these are our protein.sequences, and we download them, and then we have them, and then we want to return them because I want to make a function again. Right, so I'm just going to say protein.sec, return it, and if we didn't find anything, I just want to return a null. Return a null. Right, and I'm going to make this a function, so this is going to be query proteins. And that has that's a function, and it has an ID, right? The ID that I want to query. Standard, I want to not query the American sequence, but I want to query the other one. So let me get the one that we were looking at, which was on Firefox. So let me go to Firefox. So the Zaire version of the protein, which is called this one. I'm going to change here the ID, and then that's it. All right. So now if I would do query proteins, query proteins, and I would just call the function, then I would have m.prot, my proteins, or something like that. And again, this one goes into a new file. So I'm just going to create a new file, copy the function in, and I'm going to call this query proteins.r. And I need to make sure that in my analysis, I now source r/query proteins.r. Right, so this makes the function available, and then I can call the function. So let's try that, right? So let's make sure that we run the whole code from top to bottom, right? So just to make sure that we didn't do any mistakes or that the code doesn't depend. So I'm going to go to R, and in R I'm going to throw away all of my objects, right? So if I remove my objects, I can just do this call, and now my R is completely empty, nothing is loaded. So I'm just going to copy paste in the code, right? So we're going to download the data from all of the sequences, so all of the DNA sequences. We're going to create our BLAST database, and now we have m.prot, which should contain all of the protein sequences. Of course, let me just look at the first one. And so here we see the first protein, which is called chemokine binding protein from the monkeypox Zaire virus. Of course, it's very mean to now just use this protein and query it, right? But we can, if we want. We can just query this to the genome database and see how many hits we get. But of course, this would perfectly fit to the Zaire genome, but it might not fit to the current version of the monkeypox genome. But that's why we want to build, in the next step, a protein model. Right, so I am going to query a couple of these, right? So I'm just going to say while m.proteins, right? Because we know that from R, right? We know that if we ask for the length of this, that we have 191 proteins on the genome. So let's just download like 20-ish annotated genomes. So get the proteins from like, well, why not do all 75? We're just going to do all 75. So I'm just going to say, so for ID in the names of the NCBI sequence, right? I am just going to query the proteins of this ID, and then I'm going to remember them. So I'm going to say proteins. Oh, I'm going to say proteins is initially empty. Then we're going to query the protein database, and we're just going to say proteins. I don't know why that doesn't work. It gives me the suggestion to, but I'm just going to say proteins. So I'm just going to go through every genome that I have loaded, query the databases, and then full screen is okay. It's not your internet. It's a bit blurry when it's small. Yeah. Let me make R a little bit bigger, just to make sure, right? So say when it's good. I'm gonna go to Edit, GUI Preferences, and then I'm going to do size 12. Ooh, then it becomes really big. Well, in the end, we just want to see like fancy. Let me just say names of m.protein. Right, so these are all of them. So this should not be blurry now, because it should be like big enough to view. All right. So the thing that I'm going to do is I wrote my little query proteins function. I'm going to say I have a list of proteins. Initially it's empty. I'm going to go through all 75 genomes, and then I'm going to query the proteins associated with that genome, and I'm just going to make a big list, right? So in the end, if every genome would be annotated, I would expect 75 times around 190 proteins, so that's like 7,000-ish proteins, but not all of them are annotated, fortunately. But let's just start this in R, right? Because I am going to do one more thing, because I'm going to say sys.sleep and make sure that you sleep like half a second after querying the proteins. Just do not stress the server too much, right? Be a friendly neighbor and don't stress the Ensembl or NCBI server. Good. So, and this will run for some time, and it will download all of the proteins, and we could quit it halfway through because we don't need all of them. But in this case, we could get an idea. All right, so this is just a little bit of waiting. So I'm going to do a quick cigarette break, and I will be back in a couple of minutes, and then we can more or less continue. So let me mute my microphone. I didn't prepare any animated GIFs. I'm very, very sorry. Normally when I do lectures, I create animated GIFs and I have this like waiting screen. But unfortunately, I forgot to do that. So you guys just have to look at the background screen and I will be back in like 5 to 10 minutes. And we have to wait anyway, right? Because it's a large amount of data and we're only going to do this once and I'm going to save it to a file to make sure that I never have to download it again. Um, just to be like friendly to the NCBI server. Good, but we're making good progress. Like if we look at the code so far, we did to do 0, 1, 2, and 3. So we're kind of halfway through what I wanted to show you guys to do a real kind of analysis. And of course, all of these things are in the R course. All of these like tips and tricks like using grep or using the lapply or using apply. You learn those when you do the R course, and I think after like lecture number 5. All right, very good, very good. It actually finished, so I'm going to forgo my cigarette. So let me ask the length of proteins. Now, first look at the warnings, right? So the warnings are a lot of them, right? Because it gives me all kinds of XML errors, and that is because a lot of these don't have anything annotated. Right? So if there's no annotation in the protein database, it just gives me an error when I try to query non-existent sequences. But now when I look at proteins, I should have a big list. So what is the length of it? So we downloaded in total 12 proteins. That's not a lot. That seems too few. That seems way, way, way too few. Unnamed. That is a very interesting. That is very interesting. Because I expected it to be much, much, much longer. But that's interesting why it doesn't do that. Hmm, interesting. Alright, so let me see. Names of NCBI sec, those are these. Ah, I already understand. I already understood. We use the names of the sequences, which is the FASTA header. But the FASTA header is not what we should use the short name. So we should not have used the names of NCBI sec. We should have used the row names of our little table, which is called source IDs. So let me fix that and then row names of this one, source IDS. All right, very good. So let me check, right? So let's go to R, make sure that we check that. Um, so yeah, we only want to use this part of the name. So I'm actually surprised that it found proteins while giving it the full FASTA header. In some reason it figured it out, but that means that I can still have my cigarette because we need to download our proteins again. So I'm just going to copy this, go to R, and then paste it and be back in around, well, took around 4 or 5 minutes, right? So I'm just going to do a very short break and then we can work with the protein sequences and start doing some protein models. Good. So I will be right back. Alright, so I'm back. Let me guys show you what R came up with. So R came up with an error, and that probably means that I was querying too fast. But let's see, right? Length proteins, how many did we get before we got cut off? So we got 5,310 proteins, which I'm perfectly fine with. So this is more than enough. So we now have all of the protein sequences. We have, well, not all of them, but enough of them. And we have all of the 75 genomes that we are interested in, so we can now continue. So, very good. So next step, go back to Notepad. Create a protein model, right? So we need to now find one protein that we want to focus on. So there's two ways of doing this. We could create a protein database, take a protein and blast it. We could also just look at the names, right? And see if there's any names that strike out or are interesting to us. So let's go to R and just look at the names of the proteins, right? So this is a long list, and I'm just going to table them because the same protein should be in there multiple times, of course. And then when we go all the way back, then we see here, so there's a bunch of these GP 103s that only occur once, and we also don't really see anything interesting here, so I'm just going to look at the top 25 names after tabling them. So for some reason, all of them are only in there once, which is a little bit annoying. Let's ask for the maximum value to see if anything occurs twice. No, which is logical that they don't occur multiple times. And that is because they all have unique protein names. So that's something that we have to remember. But I wanna see a little bit more. I want to see if there's an interesting protein name that we can use. And otherwise, I'm just going to take one. So there's this thing called the virokine protein. So let's see if that is actually downloaded. So I'm just going to use grep. So I'm going to say grep through the names of the proteins. And what do I want to grep? Well, I want to grep for virokine. Why not? Right? It's just the name of a protein. So let's go to R and see if it's there. So yes, we have 7 versions of the virokine protein. So if we look at the proteins, then I think I selected this because it's not too big of a protein. So you can see that it's a relatively short protein, right? And you can see that it's in several monkeypox viruses that they found it in, and we can see here that it's not a crazy long protein. Right, and when we look at it, we kind of see that it almost seems like there's no variation in them. So that's a little bit of a shame because we would want to have a virus or a protein which has a little bit of a variation, right? If there's no variation, then we can build a consensus model. We just know that the protein is the same. But in theory, we could just take this protein, blast it to the genomes, and see where it is located in each of these genomes, and if it's located more than once in some of the genomes. But there doesn't seem to be any variation in this case, so it's not too interesting to build a model of. So let me actually do something else, right? Because I want to know which names occur multiple times. And I tried to use the table, right, which didn't work because of the initial name. But you can see that if I would just take the names of the proteins and then just do a string split on these names by space, and then I would kind of dump the first one or take this. And so now we already start seeing that there are many more interesting protein names, right? So for example, the monoglyceride lipase. Perhaps that's a little bit bigger. So I'm just going to grep for that and look if there's any variation. So yeah, here we see some variation, right? So it's MST in one version, MSA in another version. So we see that there's a little bit of variance in this protein, not a lot. But there is a known more or less polymorphism when we downloaded it. Good. So now we have our proteins, right? So we can do two things. So we can we can just blast these proteins, or we can do a multiple sequence alignment. But the first thing that I like to do is make sure that we use something which is called Levenstein distance to make sure that we know that our proteins are relatively closely related. So we can do this by using the distance function, which is from the string dist package. So when we go here, what we can do is we can just say, give me the string distance of all of the proteins versus all of the proteins. And first, the string distance of the names of the proteins, no, not of the names, of the proteins versus all of the other proteins. And I want to use method is LV. Let me just do it for the first protein against all of the other ones so that you guys can see how it looks like. So when I would do this, then it will tell me the distance of this protein to all of the other ones, right? So we see, for example, that protein number 1 has a relatively, or has no variation to this one, but it has only 6 distance to this one. So this Levenstein distance gives us a number, and this number, the higher the number, the more divergent these are, right? So if I would ask anything under 25, right, then I could ask which ones are lower than 25 in distance. Then I see that this protein actually has a lot of other proteins which are very, very similar to it. So if I would just look at proteins, and I would just look at these entries, right, then you can see that, yes, these proteins all look very similar. Sometimes it's a little bit longer, there's more variance in there. So let's just use this one, right? Why not? So I'm just going to say that my selected protein is the first one from the list. We can look at the names of them as well. So we are looking at the chemokine binding protein. And this chemokine binding protein is sometimes called slightly different. But using Levenstein distance, we can say, well, 25, we could be a little bit more stringent and see like what will happen if we have a maximum distance of 15, then we see that we still get like 55 proteins in our 5,000 protein database that we just built, which are very similar. Good, so let's make a consensus sequence out of this one. So we have 55 entries, so I'm just going to take this whole code, right? And I'm going to go to R, and in R I'm going to say, well, do this, and then say these are similar, right, to or similar to the first one, right? Of course, we could do this for all of the proteins, but then we end up with like 180 something unique proteins. But now we have one protein which is found in a lot of these different strains, and it is very similar in all of these strains. Sometimes it's a little bit longer, sometimes it's a little bit smaller. So we need to do multiple sequence alignment. And fortunately, we have the MSA library already loaded because that's here. So we could just do the multiple sequence alignment of these proteins and be done with it. So I created, I think, a little bit of code because we need to make an amino acid string set. That's what it's called. So we can say, so it's similar. So when we say proteins similar, we get all of the ones which are similar to the first one. So similar to this kind of chemokine binding protein, right? And then we just say AA string set. And then we create an amino acid string set out of it. And then we just do a multiple sequence alignment on it. Right? And then we call this my MSA, which comes out. Alright, so let's go to R. Let's run the multiple sequence alignment on these proteins. Amino acid string set. The S is also capitalized. Selecting a method. Object similar, not found. Oh, right, because I didn't save it. So let me go back to Notepad, make sure that I grep. So let me delete this one. So let's first find all of the similar ones, and then we use those to build a multiple sequence alignment tree. So we go to our GUI, and I forgot to fix the S as well. So let me just copy paste it, and alright, so now I did a multiple sequence alignment, so we can just plot this if we want it. Right, and then, oh, we can't plot it because we need to first convert it to a second error alignment. But we can get the consensus sequence, I think. So we can get the consensus sequence by using the MSA consensus sequence function on the multiple sequence alignment that we did, and then it will tell us that this is the consensus sequence. Right, so we can see that every protein starts with more or less the same, then there's a part which is variable, and then we have the end of the protein which again is very similar. So it's just the MSA. If we would look at my sequence alignment, it would look like this. So you see that there are some which have like an insertion and others which don't. So we build a consensus model, and now we can use this consensus model to scan, right? So we can use this consensus model to find where this thing is located in the monkeypox genome. So let me get this code. So my MSA, we now have our consensus sequence, and then I want to TblastN the consensus sequence to the viral genome database. So fortunately, we already built this database, so we don't have to do anything like that. So we can just use BLAST. So how do we use BLAST? Well, again, the input to BLAST is going to be a FASTA file. So we're going to do the same thing as that we did before, but this time, because I'm doing BLAST, I'm going to use a temporary file. So I'm going to say temp file is, or I'm going to call it TMP file, is the function temp file, and this will just give me a temporary file. I'm going to empty out the temporary file, TMP file, and then I'm going to say for header in. No, I don't have to do that at all because I have a consensus sequence, so I only have to blast one. So I don't have to do a for loop. I can just say cut. This is going to be consensus for, what was it called again? Chemokine binding protein. And we're going to use the consensus for the chemokine binding protein. We're going to do a new line. And then we are just going to cut the consensus sequence, and we are going to add a new line. So this is going to be my query file. So for BLAST, I need to specify what I want to get back. And I prepared this beforehand because it's just a lot of explaining to do. But when you go to BLAST and you go to the BLAST manual, then in the BLAST manual, you can say that you can specify your own output format. So how do I want the results from BLAST back? So I'm going to say I want to have it 6, which means I want to have it back in a table. I want to have the query sequence ID, the sequence which we find, the start, the end, the length, and these kinds of things. And then I'm just going to call BLAST, which I installed on my computer. So I'm just going to say, wait, we forgot one thing in the make BLAST DB. Because when we make the BLAST DB, we only output the FASTA sequences. I forgot that I also have to do an indexing step here. So the indexing step is relatively easy. I'm just going to say this. So I'm going to make a system call where I say make BLAST DB. So this is just going to be a command line call. So BLAST needs to be installed on the command line. So it is making the call saying that the in is my database, right? So the file that I have. And then I need to give it a name. So I'm just going to say this is viral genomes or something like that. And my database type is going to be nucleotide. Let's keep it the way that I had it, right? So here we have a database. Then we have to specify a name. So name is monkeydb or monkeydna. And we are going to say db type is nucle. Right, so now I'm just going to use these variables. So let me actually rerun the making of the BLAST DB in R so that it does the call to the command line as well. And now when I would have db, no, I first need to source the file to update the function. And now when I make my BLAST DB, it will actually tell me that it is building a new database. It took a couple of seconds or less than 1 second, and it added 75 genome sequences to there. Good, so now we have our database. Alright, so we have our database. So we can now start querying it. So we can say, do a system call. The tool that I want to use in this case is called TblastN, right? Because I want to blast nucleotides, or I want to blast protein sequences to a nucleotide database. The database that I'm going to use is just the database that I got back from the blast call. The out format is the format that I want to query. So I'm just going to give this format, and then I'm going to say temp file is the temp file. So this is where the query object is. All right, so let's go to R. Right, so I'm just going to first, here cut. I want to do this to file is tmp file. Append. Append is true. Right, and I'm going to have to add the consensus sequence also to the temp file, and I also want to append this. And let's just go to R, copy it in. Consensus cannot be handled. Oh, I'm forgetting to copy code again. So my consensus, and then I'm going to use here my consensus. All right, so let me do this similar, the multiple sequence alignment, do the temp file, and make it. All right, let's go back to R. All right, so now when we open up this temp file, right, which is called this, so I can just go here and get the file. So I can just say, probably here, I can go there, and then I can get this temporary file. No, I can't, because for some reason this file does not exist anymore. Well, you just have to believe me. It's just a simple FASTA file, again, which we just had. We do the same thing for the BLAST database as well. But I can't show you the file, but the file will have two lines in there. The first line will be consensus chemokine binding protein, and then it will have my consensus, the string that we just made, in there. And now we can start and try the BLAST, because we now have the file. So we just specify that we want to do BLAST with the database that we have. We want to do TblastN, and we want to specify our own output format. So when we go to R and we do this call, then it should be okay. So now we get res back, and here are our results from BLAST. So we just took our consensus sequence, and then we blasted it to the 75 genomes in the database, and with that we now get our results back. It says that here, see FASTA reader, hyphens are invalid and will be ignored around line 2. So why is it saying that? Well, if we look at my consensus sequence, you see that there are hyphens in there. And this is not allowed, because in a consensus sequence the hyphens should be replaced by ends, because anything can be there or nothing. Right? So a hyphen means a gap, but when blasting you can't have hyphens for gaps. You have to have ends for any nucleotide. So let's fix that, right? So when we print my consensus, I'm just going to say gsub. So gsub my hyphens by ends, right? So any amino acid or a gap is perfectly fine. So let's go back to R. Let's copy paste the code again and see if we still get the error at the beginning. So when we look at res now, then we go all the way to the top, and now it has no problem with this. Good, so you see that we just get a bunch of strings back. Every string is one line of the BLAST output. You see that the output is very good in some cases. Some cases it's not that good because the e-value is in the last column, like I asked. But now we have to parse this, right? So we have to parse this into a format that we can use. Right, so what are we going to do? Well, first things first, we are going to remove our temp file, because we don't need it anymore. We already have res, which contains our BLAST results. So now what we want to do is we want to make this into a matrix. And this is relatively easy, but what I'm going to say is I have values, right? The values are initially NA. So that's every value in my database. So what am I going to do? Well, if the length of what I got back let me actually move this. So if the length of the results that I got back is more than 0, right, that means that BLAST found something. Now I can get the values that I have. So I'm just going to say, well, take my results and string split it by the tab character, right? Because in R, we saw that we have this tab character separating the individual values. So that's what we can do. And then we are just going to lapply to res the string split function, and we are going to split by tabs, and this is going to be perfect because when we do this in R, then we see we get something like this. Right, so it says that the consensus was found in this ID from, and then all of these things. Right, so I'm just going to lapply the string split, and in the end, I don't care about any of this. I'm just going to say unlist everything, and those are my values. Right, so everything will now be on a single line. So if I now would look in R, and I would do this call, and I would look at my values, then the length of my values would be 2016. Why 2016? Well, I have an X number of rows. Every row contains an X number of values separated by tabs. So I have in total queried for 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So I asked for 14 things. 14. Anyway, I asked for 14 things, right? So I have 14 columns, and I know how many rows I have because my rows are the length of res. So I can now just say, well, make a matrix. Make a matrix of my values. How many rows do I have? Well, the rows are the length of res. I have 14 columns, and I can directly say by row is true, right? Because the values are row based, because I did for row 1, then I did row 2, then I did row 3. And I'm going to call this MM for my matrix. All right, just simple try. Let's go to R. Unexpected symbol in MM. Ah, right. I forgot a comma after by row. Right, so now if I look at MM, then this is a matrix of the output that we just got back. So we can check that everything's okay, but it's filled in a row-wise manner. I asked for 14 columns. It gives me 14 columns, so this should all be perfectly fine. So let's just call this res now, so override the results that we got. And now we can actually say that, well, we can add the column names, right? So we can say that the column names of res are actually the names that we just queried for. So those are these, right? Because it gives us in this order, and I need to make sure that I change this every time by this, right? Because those are the names of the columns that we queried for. So I'm just going to use the BLAST names on the matrix that I have, and that should be perfectly fine. We have mismatch. All right. Oh, that's bad because we want to do here. All right. Layout a little bit. Comma. Change it. All right, so these are then the names of the columns that we have. Right, just the same as that we had before, but now we just put them on res. All right, let's go here. Let's go to our GUI. Right, that's true because actually there was a comma missing and I didn't fix that. So let's fix that. And now when we look at res, let's look at the first 5 rows. Then it looks like this. Right, so we can see we queried the consensus to this sequence. It found from 1 to 252, so it found the whole sequence. But in the genome of the virus, it starts at 197,000 and it ends at 195,000. And we see that in the next genome, right? No, in the same genome, it finds the same sequence again, but now at a different position. So it finds it at 1700 until 1000. So this gene is actually twice in the genome. So that's something that we learn. We learn that every monkeypox virus comes with two of these chemokine binding proteins. So we already learned something. That's good. That's good. Okay, but now we need to do something else still, because we need to make res a data frame, because when we look at it in R, we see that everything is a character, which we don't want because some of these columns are numeric. So we're just going to say data.frame res, and these are my BLAST results. So I'm just going to call them res again. Good. One of the things that we know from BLAST, and let's go back here, is that every BLAST hit comes with an E value, right? And the E value tells you how good the hit was. So we see here that some of these hits are very, very certain. Like it's kind of a P value, but then for BLAST. But we see that this one actually is not really significant. So it seems to be in the genome, but there are 5 gaps and 30 base pairs in the gap. The number of identity matches is only 40, so it's 40 amino acids of the whole 142. No, of the whole 252 proteins is in there, right? So it's only a very little piece which matches, but the rest is all mismatches. So that's why it gets a very low E-value score. Furthermore, we see that something annoying has happened with the sequence IDs, right? Because our sequence IDs, which are in the row names of, how did I call that again? I called this source IDs, right? So because when we look them up, our names are like this. But for some reason, while blasting, it put something in front and in the back, right? So we have to fix the source sequence ID column, because it doesn't one-to-one match the IDs that we gave it. And this is common in BLAST. BLAST always does this. It recognizes that this is a GenBank ID, and sometimes it recognizes IDs as being reference or reference sequence IDs or other types of IDs. So it puts these things in front. Fortunately, we can easily parse this out, because when we see 2 of these horizontal bars, we know that our ID is going to be in the middle of them. So we could use a very fancy regular expression. I generally don't really like regular expressions, so I'm just going to use the lapply and then do a couple of greps to kind of get rid of that. So let's go back here. So the first thing that I want to do is I want to say res, comma, e value, right? And as numeric, just to make sure that it's a numeric value, this needs to be lower than 0.01 before I'm going to consider it. Right, and then I'm going to ask which ones have this low an E value, and then I'm going to take those. So I'm going to subset my matrix with E values which are good enough for BLAST, so that I know that I'm only looking at hits. And then I have to fix this sec ID column. So I'm just going to say, well, res, comma, sec ID. Right, so if we go back to R, just so that you guys know how this looks. So here you see that sometimes it's GB, sometimes it's EMBL, sometimes it's the reference sequence. So there's different identifiers there. So we can't just grep or gsub or even build a regular expression for this. Well, a regular expression might still work. But I'm going to do it slightly different, because I'm just going to say, well, what do I want to do with these things? Well, I want to, for each of them, right? So for the first one, I want to grep if there is a horizontal line in there, right? And if that is the case, then I want to know how many there are, because it might be that the ID that I use also uses this horizontal line. So I'm going to string split my res, and I'm going to split it by nothing so that it goes into individual letters, right? So now you see individual letters, and now if I would grep for the horizontal bar in the string split, then it will tell me 1. Why will it tell me 1? Again, because string split gives you back a list. So I'm going to make sure that I unlist this, and then it should now give me 2 positions of which there are the horizontal bars. You see that it doesn't give me 2 positions. It gives me every position. And this is because this thing here is also a wildcard. So I have to add fix this true. right? Because I really want to search for this character. I don't want to use it as a wildcard. So the third is a horizontal bar, and then the 14th is as well. Good. So now I can build up an if statement, right? So if the length of this whole greppy thing is larger than is 2, then I know that I am dealing with one of these IDs that was changed by blast. Of course, I don't want to do the if statement here, but I'm just going to copy paste this into Notepad so that we work there. Right, so we're just trying to fix this SSEC problem where it puts them in. So I'm going to do this for every sequence ID. So I'm just going to lapply to res, comma, SSEC. What am I going to do? Well, I'm going to apply a function which works by x. And now I can change this whole thing here, this res1 SEC I can just say x, right? Because I look at every entry individually. So if this is the case, so if it is 2, then I want to get rid of those, right? So how do I get rid of those? Well, I can again use string split. So I can just say string split x, right? So string split the current protein identifier that we're looking at, string split it by this horizontal line. Fixed is true, right? And then I want to lapply to everything what we get back. I want to select, and I want to select the second element, right? Because if we go here, let me show you guys that in R, right? So if we do a string split of x, and x is called res1, SSEQ ID, we call this x, right? So if I string split x by this horizontal line, then it says that the first element is called GB, which I don't want, and I want this name back, because that's the name that I'm using for all of my sequences, is the standard, more or less, GenBank ID name. Good, so this seems to work, right? So I'm going to lapply, I'm going to select the second one, and then I'm going to unlist it, because lapply and these kinds of things always need to be unlisted. And I'm just going to return this if I want to. Right, so I'm just going to return this. If there are not 2 of these horizontal bars, we might have a different type of name. So then I'm just going to not change the name, so I'm just going to return x itself. Good. So will this work? Well, let's see if it will work. So we go back to R, and now we see that indeed it ate, or hey, it selected based on the 2 horizontal lines. If it finds 2 horizontal lines, it will take the middle element, and when there is none, then it doesn't do it. And this of course becomes more pressing when we have sequences which we did not get from Ensembl or NCBI, but when we get sequences which we download from virology.org, right? Because if we download our own FASTA files, then of course we might not have an official ID. So because of that, we need to fix that. All right, so I'm going to enlist everything, and then these are going to be my IDs. So I'm just going to put this back into the column where I took them from. So I'm just going to, oh, you guys can see that, right? So let's go back. So here we have the lapply, which works. So I'm going to then say unlist all of this, and then I'm going to put this back into the column where I took them from. So in this case, that's called res SSEC. Good. So now if we would run all of the code, right? So we would query. Oh, so let's make the query again. So let's do the whole section 5. So we make a temp file, we put our sequence in there, we specify the out format, we do blast, we load in all of the values, we split them using tabs, we put them into the matrix, and then we make sure that we only take things which have a low E value from our matrix, and then we just fix the SSEC ID names. All right. So do the blast query, and now when we look at res, then now our res table should have very significant entries, and we now see that indeed we have different, well, not all of them are perfect, like this is only part of the whole thing, but that's good. So, and our sequence IDs seem to match as well. Perfect. So now we have done part number 5. So of course, part number 5, we are going to do the same thing as what we did before. So we're going to just cut the code and we're going to make a new file. We're going to put it in. We are going to put this in R and we're going to do blast.r. Alright, so now when we are in our analysis, we're going to source r/blast.r, and then I am going to make a function out of this, right? Because this is going to be a function, so I'm going to call it blast is a function, and we're going to do this, and in the end we want to return our res. So that is good. So do we want to actually, yes, because we want to give it sequences, right? Or consensus. But in this case, we are we're dealing with the consensus sequence, right? And here I actually gave it a name, but that's a little bit wrong. But this is okay. So I'm just going to call sequence, single sequence, not sequences. Do we need anything else that we want to specify? Well, we might want to specify the BLAST tool. So this is the tool. And we are going to say tool is TblastN, because we might want to reuse this function when we are going to do standard blasts, so nucleotide, or proteins versus proteins. So we now have our blast function, which we can use on a single sequence to blast it towards all of the 75 reference genomes, and then we can do something. So just blast my consensus and then call this blast.res. Good. So now we've did more or less 5 things on our list. So we downloaded our sequences, we made a BLAST database, we queried the proteins from NCBI, which we gave us 5,000 proteins. We look through all of the proteins. So we take the first protein in the list and we look to see which ones of them are similar. And when they are similar, we're going to do a multiple sequence alignment. And then based on the multiple sequence alignment, we create a consensus, and then we blast this consensus to the reference genome. Good, so now we are at a point where we can finally start making graphics. Right, so I'm going to start making graphics. So I'm going to say plot. All right, so what do I want to plot? So I first need to figure out how many elements I have on the x-axis. Right, so on the x-axis I'm going to put all of the different viral genomes. Right, so I'm just going to say plot C0, n row of my source IDs. Right, because I have that many genomes. I need to figure out what the Y range is, right? So the Y range, I could just say, well, the virus is like 200,000 base pairs, so I'm just going to say from 0 to 200,000. But I don't know if that's exactly true. Fortunately, we have our BLAST results, right? So if I go to R, then I can show you the BLAST results. Oh, I have to source, of course, the BLAST function. Right, so now when I look at blast.res, let's look at the first 5 rows. It has the S start and the S end. So what I'm just going to do is I'm just going to take from blast. Oh, something goes wrong. My notepad windows one. So I'm going to take from blast.res. I'm going to take the start as numeric, and I'm going to combine this with the sequence end, and then we're going to do it like this. And now I'm going to ask, so these are all the positions, and I'm going to say min positions, comma max positions, and this should give me the lowest value and the highest value. So just to see if it works, let's go to R. So in this case, the minimum location of the first of this consensus sequence is at 247, and the other one is at 227,000 actually. So much larger than I would have expected. But we can go here and we can now say that our Y is running from the minimum of positions to the maximum of positions, and I'm just using a comma. I'm going to say type is none, x-act is none, I don't want an x-axis, and my x label is going to be empty. Right, so when we go to R, how does this look? So it looks like this. So very basic, empty plot. We have here the positions, which is fine. We have here all of the different reference genomes, or at least space for all of the different reference genomes. And we're now going to add the names to it, right? So we're going to just say, well, add these names to the axis. So axis 1, which is the x-axis, at is 1 to the number of rows of the source IDS. And what do I want to put there? Well, I want to put the names there. But the names themselves don't really mean anything to me, right? Because I'd rather have something like USA 2020 or USA 2020 and then have my subtype on there, right? Because you can see that some of them are called Acmeta, some of them are cowpox. So I kind of want to know and see this in the plot, right? So I'm just going to say, well, I'm going to make something called display names. And the display names are going to be a paste 0 of the source IDs and source IDs. And I'm going to take the country, and then I'm going to take source, and I'm going to take the year. And I'm going to take from source IDS, I'm going to take the subtype. I think that's written with a capital. Yeah, subtype. So sub and type, everything's written with a capital. So sub and type. All right, so if I now look at my display.names, display names. So I just do the call in R, and then when I look at the display names, I see that, oh, that looks bad, right? I want to have spaces in there. But the first one is USA 2020, Monkey 22. So let's put some spaces in there as well. So I want to have a space between here, and I want to have a space here for the subtype as well. All right, let's go to R. Let's paste in the display names, look at the display names, and these are names which are suitable to put on our plot on the x-axis. So how are we going to do that? Well, we're just going to say at these positions, show the display names, don't show the original names, right? And of course I need to make my plot a little bit bigger, so I'm going to say op par mar is C. Give me like 10 at the bottom. At the left side I only want 2. At the top I don't care about, and the other axis I also don't care about. So I'm just setting some margins so that when I make my plot, the names will kind of be able to be shown. Alright, so here we see our plot then, and I have to make sure that they are rotated. So I'm going to say les equals 2, cx.axis is 0.7, right? Because they are too big to show otherwise. So just a little bit of layouting, and now we can see that indeed we start seeing more or less all of the 75 genomes that we have. So we see the different monkeypox, then we see the Nigerian outbreak from earlier, so 2017. Here we see, for example, the different Acmeta viruses, and here we see, for example, the vaccine. We here have the variolas and the Yamada strain, which are all not monkeypox but smallpox. So now we can actually start plotting the data from BLAST on this little consensus sequence across the genome. So let's do that. So let's do that, because in the end we want to kind of visualize the genome and see if the layout of the monkeypox genome, the current monkeypox genome, is different from, for example, smallpox, which would be good because we don't want it to be smallpox. But here we have our display names, we have our axis, we have our plot. So the only thing that we now have to do is make sure that we plot all of the different values, right? So what are we going to do? Well, we have a single protein that we're looking at. So I can just say for um, name in not genomes. I'm just going to say for name in the row names of source IDs, right? So now I need to find if this name is actually in the BLAST result. So I'm going to say BLAST results at this at this column that we just fixed because the column was wrong. So let me see BLAST DB, BLAST themselves, which was called sec ID, so source sec ID. Is his name, right? Which ones are those? And I'm going to call this II. So for I in II, right? Because it might be that the same gene is multiple times in the genome. So I have to make sure that I have a for loop for all of the genomes, but I also need to have a for loop for all of the times that the gene might be in the genome. And then I'm just going to say, well, okay, so now I know on my blast.res, right? And I know that I can take I, and I know that I need to have the S start, and so this is YS. The end of Y is of course the same, so S end. And now I can do a plot, and our points, right? So I can say points. Which gene am I currently at, or which row of the genome am I currently at? Well, I just have the name, so I don't know that, but I can figure that out because I can ask which row name of source ID. So my X position is going to be which row name of source ID is the name that I'm currently looking at. Right, so now I can say draw a point at xpos, xpos, and then we can say the y position is going to be y start, y end. And we are going to say type is line, something like this. Alright, let's see what we have so far. Right, so we're just going to very basically iterate over the genomes. For each genome, we're going to try and figure out if the gene that we are looking at, which we made a consensus sequence for, is in there, and then we see that it looks like this. Right, so it's very, very tiny. I hope you guys can see it. Actually, let me do this a little bit different. I'm going to say B for both. PCH is 19, and color is orange. Alright, so let's add some orange colors. Right, so good. So now we start seeing some interesting things, right? So what we can actually see is that this gene is normally 2 times in the genome of monkeypox, right? In the new monkeypox, it's at the beginning and it's at the end of the genome. However, when we, for example, look at the Central African region, so the other clade, because in Africa you have 2 different strains of monkeypox. You have the West African strain and you have the kind of Central African strain. But the Central African strains don't really seem to have 2 copies of this gene. They only seem to have 1 copy of the gene at the end of the chromosomes. If we then look at all of these here, these don't seem to have any copies of the gene. So these here are the Indian, so the goat variant of monkeypox. Right, so this is goat pox. Goat pox doesn't have this gene at all. And we see that the same thing holds for some of these other entries here, which are like the turkey pox and the ones from Marseille. And so the the bird version of the genome does not have this gene. The more or less the very virulent strain from the middle of Africa has 1 copy of this gene, and the monkeypox, the standard monkeypox, which are not that heavy, more or less, they have 2 copies of the gene. And we see that this is similar to, for example, the smallpox and the Acmeta, right? So Acmeta and smallpox are here. Smallpox seems to have 1 copy of the gene at the end, very similar to the African strains, so the Middle African strains, not the West African strains. But the Acmeta virus is very similar to the monkeypox virus, and it has 2 copies of the gene. So of course, we don't just want to do this for a single protein, right? We wrote all of these functions to do BLAST and to all of these things. So we kind of want to scale this up and not look at a single gene, but probably look at 4 or 5 different genes, right? So let's just do that, right? Because we can use these BLAST results, but we have to make some changes because doing it for one or doing it for many is of course slightly different. So let's just say, well, we create here a protein model, right? So we take our first protein, we do a model, and then we give it my consensus. So let's just do this. So for the first protein, so let's just do this a couple of times, right? So say for x in 1, 2, 5, right? Let's take 5 different proteins, make a consensus sequence, and then we need to remember. So my consensi, right? So this is going to keep 5 of these consensus sequences, and I'm just going to say C D my consensus. Right, and since we are now looking not at the first one but at the Xth one, we're just going to do it like this. So let's just run the code, see if we can get 5 consensus sequences instead of 1. So of course it needs to do the MSA every time, it needs to figure out the consensus, and of course it needs to find all of the protein distances. So it's going to be slightly heavier. And did 3, waiting 2 more. Okay, so now when we look at my consensi, my consensi. Oh crap! I didn't put the. You see here, like very small coding issues, but I am going to add my consensus to my consensi. All right. Let's go to R. Let's do the same thing. A little bit of a shame that has to do a little bit of waiting again. But this is kind of how you build it up. So you do it for 1, and then you start saying, well, what would happen if I would now look at 5 genes, or if I would look at 6 or 7, right? How would the genome structure change? And of course, we can also just zoom in, right? We don't have to look at this because, hey, of course, there's distance. So there might be that some genes are longer than other genes, which we already saw from the consensus sequence. All right, but if we now look at my consensi, right, we see that we have indeed 5 different consensus sequences of varying length. Some of them are perfect and other ones are not. I'm just going to have to give them names, right? So the names here are the names that I kind of want to give them. So I'm just going to say, give them names of my consensi. My consensi is going to be paste, and I'm just going to call it gene, or no, I'm going to call it consensus, comma 125, and paste 0, and make sure that you do an underscore on there, right? So just give it some names, so that when we talk about my consensi, they actually have names. So consensus 1, consensus 5. Perfect. So now we want to blast them, but the blast function that we wrote only deals with a single. Or does it? That's the big question. Does it or does it not? Because when we look at the blast function that we made, we have here a single sequence because we fill in the name, right? But we don't have to fill in the name. We can just say for sequence in sequences, right? So we're going to, instead of having a function which works on a single one, having a function which works on multiple ones. Of course, I can't cut consensus binding protein anymore, but I can use the name, right? So I can say names of sequences. So I'm going to say name. So I'm going to cut a greater than symbol. Then I'm going to cut the name of the sequence. And now instead of using sequences here, I'm going to say sequences at this name, right? So I'm going to create a FASTA file. Instead of having 2 lines, one for the header, one for the sequence, I'm now going to create a FASTA file which has multiple query lines in there, and each of these query lines will have, or so the first line will be the header, second line will be sequence, third line will be header, fourth line will be sequence again. Good, so blast is done. So we updated the blast function, so it should now work on multiple. So instead of blasting my consensus, we're going to blast my consensi, and then we should get back what we want. Let me see, I only updated the blast function, so I'm going to source the blast, and I'm going to do the blast res, and I'm going to say blast.res just to check. And well, let me only show you guys the first 5 columns. So here we see that it indeed blasts not only the consensus sequence 1, but also 2, 3, 4, and 5. Right, so now we have multiple positions in the genome, so we can now do multiple images or add multiple genes to the plot. So let's go back to Notepad. So this all works. So now we just have to add another for loop here. So instead of going through the names of the row names of the source ID, I want to know which gene we selected. So have which genome it fell on. Then I want to go through all of the hits, and then I want to color it. And now I want to use different colors, of course, because now I want to have the color of the node be based on it being consensus 1, 2, 3, 4, or 5. Right? So how do we figure out which one we have? Well, we can do that here. So we can say that Q for query. So our query is actually here in QSEC ID, right? Query sequence ID. So the first column contains the query sequence ID and the second one. So now if we can just cut. We can cut the query and then put a new line behind it. So if we now run the code to plot, it should print all of our query sequences to the screen, and it should actually fill them up all in orange. But we don't want everything to be orange, because then we can't compare which gene is located where. So what can we do about that? So we can just use, if we look here, we see that consensus 1, 2, 3, 4, 5. So we can just say, well, I want to string split Q. I want to do that by this underscore, which I put in. And then I want to unlist, or I want to lapply to this the selection function, and I want to have the second element because that contains the number, right? Because it's consensus underscore number. So this will give us the number. And we are going to unlist this, of course, just for good measurement. So if we would do this in R, then it would show us that the last consensus that it looked at was number 5. Good. So now we can define multiple colors. Oh, I didn't show you guys the R window. So when you just do the unlist, right? So the Q that we're looking at is this consensus 5, and this just takes the last element. All right. So now we have N, and now we need to define some colors, right? So we are looking at 5 genes. So let's just use color brewer to get 5 nice colors. So I say brewer.pal. Give me 5 colors from, for example, set 2. And I call this cols. Right? And now I have my number. So I can now say, instead of using orange, use cols, right? But use N. So which one do I want to select? Well, I want to do as numeric N. And now it should use 5 different colors, one for each of the different proteins that we built a consensus sequence for. Alright, so let's start looking at the genome, and now we start seeing more and more structure of the genome. We only look at a couple of genes here. I could actually zoom in, right, by saying that if we look at the code, I'm saying, well, we get the starts and the end positions, right, for the gene, but when we set the plot, we set it to the minimum to the maximum. But we can also say, well, I only want to see, for example, not the maximum, but I want to see the first 25,000 base pairs, right? Just zoom in on the lower half of the genome. So when we zoom in to the lower half of the genome, then we see something like this. So we start seeing genes, right? It also shows where the gene starts and ends. I don't like the both, actually. Let me actually do the plot twice. So I want to have points once, and I want to have lines. And when I do lines, I want to not have a line type. And could be like this, right? Just to update it a little bit. So instead of using the B for plotting both the points and the lines, I want the lines to kind of connect the two dots. Right, so now we start seeing that there is genomic structure. For example, this Nigerian strain from 2017 looks very funky, right? It has the same gene twice on the genome. This gene is 3 times on the genome when you look at the current monkeypox virus. But this one is an outlier here. This Nigeria 2017 sample is not like the other monkeypox viri that we looked at. We see here, for example, that we have the Nigeria 2018 sample. Here this blue protein is truncated. So it's not the whole protein, it's just part of the protein. The other part of the protein is either gone, um, but it's not because so there's something going on there. But hey, we can see now also that the more deadly monkeypoxes, they have this blue thing, or they have this purple thing, but much closer together. And we see here that, for example, the USA 2003 outbreak, they had this green gene, which does not occur into the current monkeypox virus or in the West African strain where it originates from. We can see that this green gene actually does occur in the Acmeta viruses, so in the other pox classifiers. So we can start reasoning about these things. And of course, we don't have to just look at the genome. We can zoom in much more, right? If we want to look at the first like 7,500, it's just changing the code, saying 7,500, and we just do the plot again. And when we do the plot, then here we zoom in a little bit more, and now we start seeing really structure of the genome. So we see that our gene here is actually found, the first one in the USA, and this is probably why they built version 2, right? Because this is version 1 of the genome, so you can see that there are issues, because this protein is like it has multiple stops and ends, so it's not consistent. Um, but this is what I wanted to show you guys. So hey, you now have the ability and the code. Let me commit the code to Github, by the way. I didn't do that. So I want to add git add r slash download data.r git commit -m download data git push. Alright, git add r slash blastdb.r git commit -m blastdb.r git add r slash, I think I already pushed the annotation, so we have query proteins.r git commit -m query proteins row proteins. Very good. And then the last one is blast, so git add r slash blast.r git commit -m blast.r. Very good. Blast. Okay, and then git add analysis.r. Oh. Git commit -m end of stream code. Git push. Alright, so I just updated the whole repository. So let me show you guys the repository just to make sure. So when you go to Monkey, right? Monkey 22 on my GitHub, then now we have the analysis code that I just coded with you guys or for you guys to show you. And of course we have all of the little functions that we created in the R folder. So blastdb, download data, query proteins, and in the data folder we have monkeypox annotation. So that's kind of what I wanted to show you guys, that in 2 hours we started with nothing, with no knowledge about monkeypox, and currently we have an ability to download the data, download proteins, use these proteins to annotate the current unannotated versions of the genome. We can compare our virus variant that we are currently looking at to many different other virus variants, and we can start drawing conclusions. We can start drawing trees as well. So with that, that's what I wanted to show you guys. If there are any questions, then please let me know. But for me, this is kind of what I wanted to show you guys, to show you how easy it is to use R using several packages to kind of get an idea of what's going on and do real bioinformatics, even with just following a basic programming course in R. Um, we're like we've now done from the R course, which generally is like 12 lectures, we've done like 6 lectures. And I think everything that I use today is more or less lecture 1 to lecture 5. So if you are following the current streaming course online, then you should more or less be able to follow what we did, and um, you should be able to replicate the code if you wanted to, right? So it's just starting off with an idea, knowing where to get the data. So for that, follow the bioinformatics course to know which databases there are and where you can get your information. And for the rest, we can make these really beautiful plots comparing 75 different monkeypox viruses. And of course, you can add more. If you download the FASTA files from virology.org, you can just load them in and add them to your database before you make the BLAST database. If you are thinking like, I'm a conspiracy theorist, right? Or I really would love to know if any part of COVID-19 is located in monkeypox, you can do that. Just add COVID-19 genome to the genome database, download the COVID-19 proteins, and start blasting them to the monkeypox virus to see if they're there. Right? So with this, I'd like to thank everyone who's watching, and thank you guys for being here. And if there's any questions, just hit me up in the comments down below, or go to the Github, make an issue there, fork the Github, take the code, do with it what you want. Just play around, do your own analysis, do your own research, like people say, but do it in a way that makes sense, right? So use raw data, make your own analysis, and draw your own conclusions. Good. So with that, we're more or less at the end. 2 hours. Thank you guys for watching, and I will see you next week Thursday for lecture number 6. Probably it's going to be linear regression, I think, something like that. So I will see you guys in a week on same platform, slightly different time, slightly different day, but thank you so much for watching. And just looking at chat, there's no questions. So perfect. Then catch you guys later, and thanks for being here.